Hi everyone, this is Scott Reaves, FSN 329 Vitamin A and Carotenoid Lecture, and we're going to kind of go through this again. I'm going to pick up where we left off, except before we do that, I want to add a little bit of information to this slide. We talked about how some of the carotenoids have vitamin A activity, and I also mentioned that quite a few of them don't. But that doesn't mean that they don't have potential health benefits. And so if we look at these molecules on this page, you can see up at the top that's lycopene, and lycopene is one of the more famous carotenoids, I would say, and it's red in color, and it is a very strong antioxidant. In vitro, so in lab settings, it's the strongest antioxidant of all of the carotenoids, and it also is a more powerful antioxidant than vitamin E. Now again, that's in the lab. Whether or not that's true in our cells is still yet to be proven, but it can have benefits. Lycopene has the highest concentration in tissues compared to all the other carotenoids. And we probably know it's because of its red color. Some of the better sources are things like tomatoes, specifically processed tomato products rather than raw or fresh. That processing actually helps the lycopene separate from other molecules, including proteins, and makes it a lot more bioavailable. So it's estimated, for example, for carotenoids in general, if it's in a fresh food, sometimes it's absorbed at less than 5% efficiency. If it's been processed, then sometimes absorption is something like 60 or 70%. So that can be a significant benefit. So for example, for lycopene, it would be better to consume sources like tomato paste or tomato sauce or even ketchups rather than the actual tomatoes. Now some studies have suggested that lycopene may be protective against certain types of cancer, but still some ambiguity there, but overall we can say it might have some good health benefits. Canthaxanthin does not have vitamin A activity. It may have antioxidant properties that are helpful. But if we look at lutein, lutein's another famous one because some studies suggest that it is protective against macular degeneration. And the macula is a portion of the eye that's in the back of the eye, and it tends to be affected by age, specifically in some people. And so that macular degeneration can be a source of a type of blindness as we get older. Um, it, Lutein is found in its highest levels in the eye, so it's there for a reason, and it seems to inhibit oxidation of cell membranes. So it may decrease UV-induced eye damage. Now, years ago, there was some studies that said, oh, lutein is beneficial for that, decreases macular degeneration, and so it got some popularity in supplements especially, and some of the press and the media really picked up on this, and now some more recent studies have indicated that there's some ambiguity there as well, and it might not be as beneficial as we thought. So the jury's still out on lutein. And the last item here is retinol palmitate. We've talked about that one. That one is obviously converted to a retinoid when that palmitate is released. Okay, so now let's catch up where we were. Let's see. We left off here, and remember, one of the things that we talked about was how important the liver is for vitamin A. It tends to be the major storage location of vitamin A in the body, specifically the retinol esters, and it's estimated that about 50 to 85%, 50 to 85% of total body retinol is in the liver, and more than 90% of that is in the form of retinol esters. Okay, so it's very important that that retinol can leave the liver as it's bound to retinol binding protein. And we talked about the significance of the synthesis of retinol, and we said that it is affected by calories, protein, and specifically zinc for its synthesis. And so that inadequacies in any of those nutrients would cause retinol to be compromised and retinol transport to other tissues to be compromised. Okay, now these are kind of the four different categories of functions of vitamin A that we're going to look at. We're going to start with cellular differentiation and protein synthesis, and then vision, a couple different aspects of vision, and then development and immunity really quickly. The first one, cellular differentiation and protein synthesis. Now, retinoic acid is the most important form of retinoids for affecting gene expression, so you want to remember that. Retinoic acid forms of vitamin A regulate gene expression, and that can affect amounts of protein synthesis for a lot of different proteins. Now, because of this, specific cell types can form, and those cell types are dependent upon adequate amounts of vitamin A and the retinoic acid derivatives. So for example, cells lining the epithelium are composed of several different cell types, including goblet cells, and goblet cells' main function is to secrete mucus. So a lack of vitamin A in the form of retinoic acid, remember we can consume vitamin A as retinol esters, becomes retinol, then it becomes retinal, then it becomes retinoic acid. That retinoic acid can affect the formation of these mucus cells. So if we look, and one of these early studies was done in the 1920s in rats, where they did microscopic examination of epithelial tissues, and they saw mucus secreting cells, and they noticed that in the vitamin A deficient rats, the normal structure and function of the cells in the mucous membranes were impaired. And that epithelium is in lots of parts of our body, on internal tissues, on external tissues. It's part of the eyes as well, and so vitamin A can affect all of those tissues. Now, if we look at vision, there's a couple different ways that vitamin A is involved. It's required in the eye in two distinct forms for two distinct processes. The first one we're going to talk about really quickly is about vitamin A in the form of retinoic acid, and that it maintains normal differentiation of the cells of the conjunctival membranes. And the conjunctival membranes are the ones on the inside, for example, of the eyelid. Those membranes secrete mucus and protect the structure of the eye. Okay. It affects the cornea, which is the transparent covering on the front of our eye, and other ocular structures. Therefore, retinoic acid prevents xerophthalmia, and xerophthalmia means dryness of the eyes due to decreased mucus. Okay? Xerophthalmia is a major cause of blindness in children in Africa and Latin America. The peak incidence of blindness due to xerophthalmia is 3 to 5 years of age, and when organizations treat children with dryness of the eyes or xerophthalmia, the core of the treatment tends to be vitamin A and a high-protein diet to compensate for both vitamin A and the ability to synthesize retinol binding protein. Now, if it's due to vitamin A deficiency, there's other things that can cause xerophthalmia and eye dryness. If it's due to vitamin A, though, it tends to come in stages. And the first stage is the vitamin A deficiency causes night blindness and conjunctival cirrhosis, or membrane dryness. That's the first stage of the eyes being affected by vitamin A deficiency. Stage 1 is night blindness and conjunctival cirrhosis, or dryness of the eyes. The second stage is corneal cirrhosis. This is when the cornea actually becomes dry and its structure is being affected. Stage 3 is called kerato, so K-E-R-A-T-O-M-A-L-A-C-I-A, keratomalacia. That means softening of the cornea. That's when that's in the later stages, and that can be irreversible at this point. So the upper right portion of this slide shows an eye that's been affected by keratomalacia. So it can be irreversible at this point. So the retinoic acid is very important for regulating undifferentiated cells to become differentiated mucus secreting cells. So if vitamin A is deficient, those cells are not made, they do not secrete mucus, and the physical structure of the eye becomes compromised. Okay? The second part of this, vitamin A's role in vision, has to do with 11-cis retinal. That functions in the retina in transduction of light into the neural signals necessary for vision. So this slide is made from a diagram in your textbook, and it goes through this process of how 11-cis retinal is involved in the vision cycle. So if we start up at the upper left, it says number 1, the light hits the retina on the back of the eye. So the retina is a layer of tissue in the back of the eye. And the second one, it says when light hits the retina, rhodopsin in the rod cells is transformed and signals are sent to the brain. Okay, so the light goes through the cornea, through the eye, strikes the retina, goes into rod cells. And the rod cells are the cell type that is involved. One of its primary functions of rod cells is to allow us to see images and movement in dim light. Cone cells are more involved in color vision, but the rod cells are involved in shape and movement in dim light. So the rod cells have a molecule called rhodopsin. And rhodopsin is a combination, so now I'm down here, of opsin, which is the protein, and this says cis-retinal. This is 11-cis-retinal, so you want to remember that specific form. When 11-cis-retinal, so this is a form of retinal, right? It's not all trans, because the bond, the double bond between carbons 11 and 12 are cis rather than trans, so it's called 11-cis-retinal. Now, that retinal binds with opsin to form a molecule called rhodopsin. When light strikes rhodopsin, it changes the form of vitamin A from cis to trans. That releases from opsin, so that now it's separated. So now we have opsin and trans-retinal. Okay? The trans-retinal leaves the rod cell, is converted back to cis-retinal, and then it can bind back to opsin to form rhodopsin. So this is basically a cycle that when rhodopsin changes its shape and loses the cis-retinal, the change in shape of rhodopsin causes a signal to be initiated along the plasma membrane of the rod cell, which moves to the optic nerve, which goes to the brain to create a visual image. So light comes in, strikes the rod cells, causes rhodopsin to change. That causes signal on the plasma membrane of the rod cell to move along its surface into the optic nerve and then into the brain to form a visual image. Now, if a person is low in vitamin A, when this happens, it takes longer for the cis-retinal to be available to bind opsin to form rhodopsin again. So what will happen is, if, for example, a person is trying to see something at night and there's a small amount of light that's forming this, the light is not able to activate rhodopsin because not enough of the rhodopsin has 11-cis-retinal. Okay? And when it does strike it and it has to be converted back, vitamin A deficiency can slow the conversion of trans back to cis-retinal. And so you don't have replacement fast enough, and so the image is compromised. So in that case, we say that they have night blindness. Another way of looking at it is if you've ever been in a dim room and a bright light flashes into your eye, that will bleach your vision for a few seconds, right? And then as you get used to it, to the dim light again, what's happening is your rhodopsin molecules are reforming. Well, if a person has vitamin A deficiency, that takes longer for that to happen. So that's a clear indicator of vitamin A deficiency. Now, if we look down here, what's happening, the left part of this shows the 11-cis-retinal molecule. So here's the kink in the structure, and it's actually bound to a lysine residue of the opsin protein. So the rest of the opsin would be over here. One of the amino acids of opsin is a lysine residue, and it's actually bound to the 11-cis-retinal. So when light hits the 11-cis-retinal, it releases from the lysine and the opsin. So rhodopsin is broken down. Okay? Other functions of vitamin A. Cellular differentiation, protein synthesis. Well, retinoic acid isomers are required for protein synthesis at the transcriptional level. So retinol enters cells and is changed to various retinoic acids. The retinoic acid binds to cellular retinol, retinoic acid binding proteins, and retinoic acid is then transported to the nucleus where retinoic acid binds nuclear retinoid receptors. So there are nuclear receptors for retinoic acid. There are retinoic acid receptors, and there's retinoid X receptors. We talked about those with vitamin D. Now these receptor complexes combine retinoic acid response elements to affect gene expression. So rather than a vitamin D response element, it is a retinoic acid response element. Okay? And the next page, or the next diagram, shows this, and I'll simplify this if we could. So this is pretty much any cell in the body. Here's the plasma membrane, and let's say, for example, this cell has taken up retinol. Well, the retinol can be converted to retinal, and retinal can be converted to retinoic acid. So most of the time you'll see retinoic acid inside cells rather than traveling around through the body. But what happens is, in the cytosol of these cells, the different forms of retinoic acids, and there's a lot of them. For example, there's all-trans retinoic acid. It binds cellular retinoic acid binding protein that moves it into the nucleus, and then the retinoic acid binds a nuclear receptor. That nuclear receptor can interact with another nuclear receptor to form a dimer. That dimer binds retinoic acid response elements. Right? These are receptors that interact with other transcription factors that affect RNA polymerase recruitment and affect gene expression. And there's lots of genes that are regulated in this way by vitamin A. Genes that are involved in cell differentiation, right? For example, immature cells becoming mucus cells, cells of the immune system, cells that are required for growth. Lots of different processes are affected by this. Now, this RXR can also bind VDR. And when VDR and RXR bind, they can also affect gene expression. So some proteins are affected by this. So for example, osteocalcin, we mentioned in vitamin D, osteocalcin's gene has a vitamin D response element. So when VDR binds to RXR, it affects and increases synthesis of osteocalcin. Now, one of the things to keep in mind is, let's say, for example, a person has too low of vitamin A or too high of vitamin A, it affects gene expression. And in the case of this, if a person has way too much vitamin A, it can actually, those nuclear receptors can kind of overwhelm the vitamin D system, and it would decrease osteocalcin synthesis. So one of the symptoms of vitamin A toxicity, of having too much vitamin A, is softening of the bones, okay, because of the alteration in gene expression. And there are some other reasons for toxicity as well. So it's very important to have the right amounts. Another case of toxicity has to do with vitamin A and vitamin A analogs, or molecules that act like vitamin A, can regulate gene expression during embryogenesis. And so vitamin A can be teratogenic, and a teratogen is a molecule that causes DNA alterations and birth defects. So it's important that pregnant women have the right amounts of vitamin A and do not take too much vitamin A and do not take molecules like or medicines like Accutane and some of the other ones that actually are taken sometimes to decrease acne or skin problems because they can be teratogens. So it's important to have the right amounts because that helps modulate changes in gene expression. Okay? So if we look at this, I just mentioned some of this. For functions of vitamin A in development, many genes involved in embryogenesis are regulated by retinoic acid receptors. Vitamin A deficiency or excess during embryogenesis can be teratogenic or lethal. Growth throughout the life cycle is also affected by vitamin A status. Like, for example, kids that are vitamin A deficient have compromised growth. Another thing, cell differentiation is dependent upon retinoic acid regulated gene expression, and that works through these nuclear receptors. So you'd want to remember that, retinoic acid works through nuclear receptors to bind the retinoic acid response element that is present in the promoter of some genes. For example, the osteocalcin gene or other genes that are involved in cellular differentiation would be good candidates to be regulated. Now, it's also important for immunity. Vitamin A, as researchers in the 1920s first recognized that vitamin A deficiency is associated with decreased resistance to infection. So it's important that that's considered in certain populations. It appears to compromise cell signaling within immune cells. Vitamin A replenishment may quickly restore immune function. So it's this kind of goes to the next stage. So vitamin A is an important part of cells of the immune system being able to function. Now, what about toxicity of vitamin A? Well, high doses ingested over a short time, as well as chronic intakes at lower doses, may lead to hypervitaminosis. So, for example, 10 times the RDA repeatedly can be problematic. So it's important that the person realizes there is a UL for vitamin A. And the UL for adults is 3,000 retinol activity equivalents. So it's not that high relative to the RDAs of 700 and 900 for men and women. Now the UL is for retinoids, not carotenoids. Okay? Vitamin A toxicity is usually due to pharmaceutical preparations of vitamin A analogs, as I mentioned before, like Accutane or some of the other ones. Now, why is it toxic? Well, one reason for toxicity is alterations in gene expression that are abnormal. Second reason for toxicity, retinoids may insert into cell membranes and destabilize them, and that can cause some of the toxicity effects. Okay. Symptoms of toxicity: anorexia, dry itchy skin, alopecia, which is hair loss, coarsening of the hair, ataxia, we know that's incoordination and movement, headaches, bone and muscle pain, conjunctivitis, and others. So there's lots of them. Again, lots of genes are affected. High beta-carotene intake is not toxic, but will produce yellowing of the skin, and that's not considered to be harmful, although it's not optimal. The Institutes of Medicine and the Food and Nutrition Board don't recommend carotenoid supplementation. If you do take vitamin A, if it is required, it should be in the retinoids. It's going to be a retinol ester or retinol acetate form probably, but not the carotenoids. Especially, I mentioned that study previously that said, carotenoids may be harmful to individuals that smoke. So they had to stop that study, so I don't think they know enough about carotenoid supplementation to be able to recommend it. Deficiency of vitamin A. It is rare in developed countries, but it can happen. It's more common in undeveloped countries, and it can cause night blindness, compromised immunity, and decreased growth. And it's estimated that each year 3 to 10 million children become xerophthalmic. 3 to 10 million children become xerophthalmic, and between 250,000 and 500,000 children each year go blind because of vitamin A deficiency. So it's a significant health issue in developing countries that needs proper attention and implementation of strategies to reduce it. Okay, vitamin A deficiency is also associated with increased risk of death from infectious diseases such as measles. So in the U.S., for example, in developed countries, if a kid gets measles, it's usually, pretty quickly moves through their system and they can recover. But if a child is malnourished, including vitamin A deficient, even measles can become lethal. So how do we assess this? Well, assessing vitamin A status is a few different ways. One is you can look for clinical eye signs or changes in eye structure, and that takes some training to be able to recognize those. But they actually physically look at the membranes of the eye to determine whether or not they might be vitamin A deficient. Another strategy is they actually ask them questions about night blindness and their ability to see at night, because that can be an indicator. And a lot of times they measure serum retinol binding protein, or serum retinol is another way that they do it. The problem is that is affected by recent vitamin A intake, so it's not a good long-term indicator, but it can often be used as a measure. Okay? And this, I included this because it kind of shows you that here's a classic example of a nutrient that's required in optimal amounts, but too much or not enough can either be problematic. So I'll let you look through this. Too much, for example, in excess is teratogenic. You can have fetal malformation, spontaneous abortions. Acute toxicities can cause the problems that are listed here. Chronic toxicities, so longer term high intake of vitamin A can cause all of these. That can lead to death. Deficiency symptoms that are mentioned here, or shown here, can eventually also lead to death. So that concludes vitamin A. So I'm going to stop this, and I hope that gave you all the information you'll need. Thank you.